So today's session is about monocots and eudicots and their applied aspects. In this session, economically important monocots along with few examples will be included, where the eudicots and the major groups like Asteris and roses will also be included. In addition to this, variation in flowers and the floral differences in monocots and eudicots will also be covered. Now, the monocots are plants which are differentiated from eudicots by having only one cotyledon in their seeds. Many economically important crops like wheat, maize, rice, and barley are all monocots. Flowers of monocots are pretty much different from eudicots. If you look here, these are actually the male flowers of corns which are known as tassels, and these tassels bear a lot of pollens because monocots are actually dependent upon wind as a major factor of pollination. And this is actually known as corn ear, which is the female flower of corn which produces fruits. And this red structure is technically known as corn silk, which is actually corn stigma. And this corn stigma receives pollens from the male flowers, and then pollination and fertilization takes place. In some monocots like arum lily, it is hard to differentiate in the sepals and petals, and the collective structure is known as spathe, which encloses this yellow structure which bear pollens, and it is known as spadix. In some monocots like birds and paradise, again it is hard to differentiate in the sepals and petals, and the combined term tepal is used. In some monocots like canna lily, the flowers look like eudicots. Now, the eudicots are major groups of flowering plants which are differentiated into asterids and rosettes. And the differences between asterids and rosettes is in the arrangement of petals. As you can see in these slides, these are all daisies, and these daisies bear petals, and these petals are actually fused at the base. Similarly, petunia is another example of asterids, which is bearing petals which are fused. In contradiction to this, rosettes produce petals which are not actually united at the base. Few examples are wax flowers, garden nasturtium, and rose species. Here, further, as an example of monocots and eudicots, coriander and wheat is used. Now, there exist differences in the roots, in stems and leaves of monocots and eudicots as well. Roots of many monocots are fibrous, while eudicots bear roots which are actually the taproots. Stems of monocots remain soft throughout their life in most of the cases. However, in eudicots, initially it is soft, but with the passage of time, when the wood formation takes place, it becomes hard. Now, there exist a lot of differences in the arrangement of vascular bundles of monocots and eudicots as well. If you look at the internal structure or the cross section of monocot stem, you will find it very hard to differentiate in the cortex and the vascular and the pith region, because actually there is no differentiation, because these vascular bundles are actually scattered throughout the stem. However, in the case of eudicots, if you see the internal structure of stem, you will find that there is a definite cortex, vascular and the pith region. Now, this is whole cortical region, and this is the vascular region which bears the vascular bundles, and towards the center of the stem is pith. In addition to this, leaves of monocots are also different from eudicots. Now, if you look at this leaf, this is a particular example of many monocots or grasses. In this case, if you see, these monocot leaves have vascular bundles or the veins which are running parallel along the leaf surface, and this kind of venation is known as parallel venation. In contradiction to this, in the case of coriander or many eudicots, there is actually one midrib, and from this midrib arises secondary or tertiary branches, and this kind of venation is actually known as reticulate venation. Further, flowers of monocots like wheat, they are different from eudicots, as you can see here, and the fruits are also different. And if you look at the seeds, you can see that wheat is having only one cotyledon, while the coriander is having two cotyledons. Further, the pollen grains of monocots are monocolpate. Like in the case of aloe vera, you can see that they are monocolpate, while in the case of eudicots, they are mostly tricolpate. If you look here, these are actually the floral structures of aloe vera or the tepals, and aloe vera is another example of monocot. If you look at the flower shape and the tepal structure and the monocolpate pollens, they are all examples of monocot flowers. While in the case of petunia, if you look at the sepals and the petals and the filaments and the carpels, they are pretty much different from the monocot flowers. Now the structure of sepals look like leaves. They are bearing epidermis on both sides, upper epidermis and the lower epidermis, and in between these epidermises are vascular bundles which are present in the region, and that region is known as mesophyll. In the case of petals, the difference from the sepal is that petals are actually the organs of flowers which are meant for pollination, so that's why they store pigments, and most of the time the pigments are stored in the vacuoles if they are water soluble. So in the case of sepals, this pigment is missing. However, in the petal cells, you will find this pigment stored in the vacuoles. Further, the petals epidermis also bear these glandular structure which receive fragrances, and they release certain aromatic compounds which attract pollinators. Similarly, in the case of carpel and anther lobes, you can see that they are also examples of eudicot flowers. This exercise will help you to further clarify your concepts of monocots and eudicots. Thank you.